طيب بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على رسول الله الأمين. Welcome everybody in this second lecture for me in this biochemistry course, the enzymes, properties, classification, kinetics, and the inhibitors of enzymes. I used to start with the introduction of the specific learning outcomes or objectives which should be covered by the end of this activity. So we'll be able to describe the classification of enzymes with some examples. This is the first objective. Second objective, to explain the mechanism of action of enzymes and the different theories regarding how the enzyme perform the activity of accelerating the reaction, and describe the coenzymes with some examples and explain the cofactors affecting enzyme activity, and describe the enzyme inhibition and its clinical significance. So let's start by this introduction. Enzymes, as you may know from your general knowledge, that they are mainly types of proteins that increases the rate of reaction by lowering the activation energy. Usually to have an chemical reaction to take place, it needs some energy either to be used for the initiation of the reaction, and this is known as activation energy. For example, to combine the glucose and fructose in the sucrose disaccharide, it needs some energy to facilitate this linking or this bonding of the two monosugars to form the disaccharide. So the enzymes mainly increase the rate of reaction or accelerate the reaction by lowering the energy required for this activation of the individual molecules of the reaction. For example, the glucose need to be excited, and the same way also the fructose need to be excited to a specific level of the excitations that can go into the reaction. The enzymes usually catalyze almost all types of the chemical reaction in the body or the living cells, whether animals, plant, or human. It's well known that like normal chemical catalyst, enzymes are not altered or change dramatically or significantly in their shape or the type of chemical nature, and they are not consumed. They can be used without changing their nature and without being consumed during the chemical reaction. So we call it reusable or turnover of the enzyme that can be effective in a specific reaction without having its specific nature change or altered. The enzymes classification, it will be later in a different slide because I have changed slightly the presentation to meet your requirements. The enzymes are not to be accelerate the reaction by a factor can reach up to 10 to the power 6 or more. That means 1 million times, or sometimes by a factor can be 5 million times, 6 million times, and so on. So it is so very, very, very big activation. The enzyme specificity and the selectivity, we know that the enzymes are very selective and very specific for the reaction. For this example this diagram, you can see my pointer. The enzyme with the plane shape of the rectangular shape, and the substrate has this tetrahedral shape. So in this example, although the atoms 1 and 4 are identical in the substrate, both the red circles of this tetrahedral shape. This looks almost the same, but usually the atoms 2 and 3 will bind to the enzyme active site, and automatically number 1 will be the third atom which will bind to the active site of the enzyme, and not fourth, because this is the specificity of the enzyme. So because of the shape of the orientation of the active site, which mimic the substrate binding specification or specific atoms, it will give this specificity of binding. So as we said, all examples or enzymes are proteins, or almost they are all proteins, except these are the exceptions, or this exception includes the ribosomal RNA and the handful RNA of the molecules that mutate or muted or mute the endonucleases and or nucleotide degaze activity known by collectively by ribozymes. So the ribozymes, although they are not protein in their nature, they are more nucleotide polynucleotide chain, polynucleotide chains. They are the only exception of the enzymes that can be of protein nature. So this type of specificity, that they are almost all proteins, or they might be some type of polynucleotide chains of the ribozymes, which have a specific role in the RNA genetic code transcription and translation. So also, in addition to the high efficiency of the enzymes in accelerations that are accelerating the rate of reaction up to 1 million times or more, they are extremely selective to their specific substrate, as was mentioned in this example. Not only to the substrate, but also the type of the reaction. For example, the oxidation reduction reactions, enzymes will not accelerate any other type of reactions. For example, the transfer of a group of active substance from one substrate to another. The classification of enzymes, in the past, it was used by just giving a specific name and with the substrate A's, identifying the type of the reaction. For example, sucrase, the enzyme which work on the sucrose enzyme disaccharide, and lactase, which splits the lactose into glucose and galactose. But the International Union of Biochemistry had made some sort of classification of enzyme according to the nature of the reaction they catalyze, and give them type of coding of the enzymes. So they are classified to 6 major classes. The first class is oxidoreductases, the enzymes that activate the oxidation reduction reaction in the different part of the body tissues. And the second class is the transferases. These are the enzymes that activate the transfer of some functional group from one substrate to the other. The hydrolases, enzymes that activate the splitting or breaking down or hydrolyzing substances using the water as the factor of the hydrolysis by adding water molecule to split the different bonds or different polymers to the specific monomers, and the lyases that split the bonds between the different part of the molecule or break down the molecules into 2 different substrates, or breaking down without using the water or by other means. The isomerases are the type of the enzymes that activate the transformation of one isomer of the substrate to the other form. For example, the alpha amino sugar to the D amino sugar, for example, or the the L sugar to the D sugar, or the L amino acid to the D amino acids. The last class, the ligases, the enzymes that link between the different parts of the substrate to form a more complex compound. I will give some examples. Like the first example is the first class, the oxidoreductase. As we mentioned, this class activates the actions of oxidation and reduction, and usually these actions are used to take place simultaneously at the same time. Whenever there is substance have been oxidized, another substance will be reduced. Example of this is the catalysis of the alcohol dehydrogenase, and this reaction is activated by oxidoreductase enzyme known as the alcohol dehydrogenase, which will change the alcohol into the aldehyde form. So it has been oxidized from the alcohol to the aldehyde. The hydroxy group of the alcohol have been oxidized to the aldehyde group. And this enzyme, for example, the alcohol dehydrogenase, is changing the alcohol from its normal hydroxyl group. In addition, in the presence of the NAD plus, nicotinamide dinucleotide oxidized form to the aldehyde. This is a oxidized form of the alcohol, and the NADH will be reduced. So this type of reaction is known as oxidation reduction reaction. Alcohol has been oxidized to aldehyde by the aldehyde as alcohol dehydrogenase. The nicotinamide dinucleotide, which was in the oxidized form, has been reduced to an NADH by removal of the hydrogen ion. So this is oxidation reaction, oxidation reduction reaction. In the biochemistry, apart from the naming of the enzymes, you can find the reactions of oxidation and reduction can be referred to as the redox reactions. So again, just type of reducing and making the name more acceptable and easy to follow. So instead of starting by oxidation and reduction reaction, in oxidoreductase as a class of enzymes, you can find the type of the reaction known as the redox reaction. So indicates the same, because whenever you have oxidation reaction, there must be type of reduction of the other substrate. So this is the first class, an example for the first class. Example for the second class is the transferases, where the class of this enzyme will transfer specific group, functional group from one substrate to another substrate. Like this example, the hexokinase, one of the very familiar enzymes which you come through in the biochemistry, especially in the glycolysis of glucose, whether the aerobic glycolysis in different part of the tissues, or the pentose phosphate pathway, or the glycogen synthesis for the storage of the excessive glucose. So the hexokinase is type of transferase enzyme which transfer the phosphate group from the ATP to the hexose sugar, for example, the glucose in this case. So if we assume this is glucose, the hexokinase will add the phosphate group by taking from the adenosine triphosphate and to attach it or add it to the hexose sugar, which in this case we assume it is the glucose. So glucose plus ATP in the presence of the hexokinase can give the glucose 6-phosphate plus the ATP, where it has a phosphate group has been removed or transferred. So this type of enzymes is known as transferases because it transfers the phosphate group from the ATP to the glucose, sugar, or any hexose. It can do the same for the fructose, galactose, or any 6-carbon sugar monosaccharide, glucose, fructose, galactose, and so on. واضح يا شباب؟ Example of the third and fourth class, the hydrolase and the lyases. The hydrolase, as I mentioned, it is a class of enzymes that activates the action of splitting or hydrolyzing or cutting the bonds between the complex molecules to the simplest form by adding the water molecule. So it's known as rehydration of the molecule and splitting it into two molecules. While the lyases, it is again type of enzymes that activate the splitting or cutting of the bonds or cutting the complex molecule into smaller molecules but without the addition of water. So both enzymes classes activate the splitting of the different, if you can name it as complex molecules, to the simplest form. These are two examples in this rectangular at the bottom. The acetylcholinesterase, it is hydrolase enzyme that activates the transformation of the acetylcholine by breaking down into choline and acetate molecule. So the acetylcholine is an ester between the acetyl or the acetate and the choline molecule. So acetylcholine is an ester. The acetylcholine esterase is the enzyme which will split the acetylcholine by the addition of water into the choline molecule and the acetate molecule. So cutting this ester bond by addition of water is activated by the hydrolase enzyme, the acetylcholine esterase. If we look at the other example, which is the aldolase, again it will split one complex form into two different substrates, but without adding the water molecule. For example, the fructose 1,6-biphosphate will be split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. So the aldolase is an enzyme that split the fructose 1,6-biphosphate into two 3-carbon molecule atoms or 3 atoms carbon atom molecules. The first is the aldehyde form, the glyceraldehyde 3-phosphate, and the other is the dihydroxyacetone phosphate. So this is aldolase. Both enzymes, the class of enzymes, they split the complex of either the acetylcholine ester or the fructose molecule. The first one by adding the water molecule, so it's hydrolase from the hydrolysis, and the other one is the lysing or splitting the complex molecule into its simplest form. واضح يا شباب؟ The same for the isomerase, will change the isomer from one form to the other form, and the ligase will link, for example, the glucose and the fructose to form the sucrose, and so on. So the nomenclature of the enzymes, as we mentioned previously, was by adding the suffix "ase" to the type of reaction which is the enzyme catalyzed or substrate. For example, the lactase act on the substrate lactose, and it will change it into its original form, which is glucose and galactose. But the International Union of Biochemistry has specify this type of more systemic classification of enzymes, and as we mentioned, into 6 groups. And this is an example of the first group and how it will be more specific to identify the enzymes at the different levels. So the International Union for Biochemistry and Molecular Biology initiate this type of naming by giving the EC, which stands for enzyme class or enzyme code, and followed by 4 digits. For example, if we want to know the naming of the alcohol dehydrogenase enzyme, which we just mentioned in the previous slide, its code is EC1111. So this type of coding stands for the first one here, EC for the enzyme class or enzyme code, and the first one stands for the first class of oxidoreductases, as we mentioned. If it is transferase, will be 2. If it is hydrolase, will be 3, and so on. So this is the first class. The first one here stands for the main class or the major class of the enzyme class, which is oxidoreductase. And the second one stands for the type of the group which is being activated, acting on the CHOH, the hydroxyl group, which is attached to any type of the aliphatic group. The one number 3, this one stands for the enzyme class that is activated either by the NADH, NAD, or the NADPH as acceptor of the hydrogen ion. So the first one will be for the NAD, the oxidized form, NADH, NAD, which will be reduced to NADH by accepting the proton of the hydrogen, and this is the alcohol dehydrogenase, which is activated by the NAD. The other type of the alcohol dehydrogenase, which is activated by, or the acceptor is NADPH, is given the last digit as 2 instead of 1. So you can go to that level of the specific enzyme which activates the specific oxidation reduction reaction. If the code is, for example, 112 after this one, it will be with cytochrome as acceptor, and if it is 3, with oxygen as acceptor of the hydrogen ion, and so on. واضح يا شباب؟ So the oxidoreductase can be classified into this is a major class, this is a subclass, and this is a subclass, and the last digit, number 4, is for the specific enzyme. أي سؤال يا شباب؟ هذا النيمينج والكلاسيفيكيشن واضح؟
الأولد نيمينج ده بنستخدمه؟
هو الروتينلي بيستخدم في التفاعلات في الرياكشنز، لكن official names which are very specific and systemic to identify the details of the enzyme and how to which class and which subclass and which subclass it belongs. This is known as a systemic name and used usually in the scientific areas where, for example, in research, for example, or when you are doing enzyme kinetic reactions, or trying to see the effect of enzymes on some type of drugs or whatsoever, you will go to that level. But usually in routine, we use a normal substrate with the ase suffix. Jamil?
I didn't understand the numbers well.
Okay, I will repeat. This coding system is a more systemic and more like organized way of naming and following the specific type of reaction. So the EC stands for enzyme class or enzyme code, and the 4 digits stand for the following, as I will mention. The first digit, now it is 1, it can be 2, 3, 4, 5, or 6 maximum, because we have got 6 major classes. So the first digit stands for the major class of the enzyme. Okay? Within each class, major class of enzyme, like the oxidoreductase, there are subclass like this type of the example, the oxidation reduction reaction, which work on the hydroxy group only, is known as the subclass of the oxidation reduction reaction. So this is a subclass, and the third digit for the subclass, which is the branch from this subclass, and the last one is branch. So each one is branch from the previous one. This is the major class, this is subclass, and this is subclass, and this is a specific enzyme. In other words, any enzyme working in the system, in the cells, will not go beyond that 4 digits. You will end up at identifying the specific enzyme with its subclass, where the first digit for the major class, and the second for the subclass, and this for the subclass, and this the last one, for example. This is the example. For example, this code stands for, the first digit is for the first class, which is the oxidoreductase class of enzyme. So this one stands for the enzyme that activates oxidation reduction reaction. If this enzyme, for example, play a role in the transfer of any functional group from one group to another, this first digit will be 2, and if it is type of hydrolyzing enzyme, will be 3, and if it is the type of, for example, lyase enzyme, it will be 4. If it is isomerase, it will be 5. If it is, for example, a ligase, it will be 6. So this specific enzyme, the alcohol dehydrogenase, belong to the first class of the enzymes, the first major class, which is oxidation reduction reaction activating group of enzymes. We have oxidoreductase. The second digit, this one, stands for the subclass of the oxidation reduction reaction activating enzymes. This specific group belongs to the group which donates the hydroxyl group, or work on the hydroxyl, or the hydroxyl group which attached to any carbon atom in the reaction. Then the digit number 3, the sub of this subclass, belong to the type of the oxidation reaction which is facilitated by the acceptor of the hydrogen ion. It can be either an NAD oxidized form, or the NADP oxidized form. So these are the acceptors of the hydrogen ion, which is split from the specific type of the substrate. So for the aldehyde alcohol dehydrogenase, the acceptor is the NAD. So this is the full code of the alcohol dehydrogenase, which is activated by the NAD as acceptor. You can find aldehyde alcohol dehydrogenase, which we use the NADP as the acceptor, the oxidized form, and this will be class subclass 2, and it will follow like this. If we are going to talk about the subclass number 2, which is subclass number 2, which is the activated by the cytochrome group as the acceptor of the hydrogen ion, then it will be 1, 1, 2 in this third digit. So this will be 2. It stands for the cytochrome as acceptors of the hydrogen ion molecule, which is being split or from the substrate. And the class which activates the oxidation reduction by the oxygen as the acceptor will be class number 3, or subclass number 3. So this will be, for example, the other type of class, and so on. The subclass can be the type of enzyme that activates the aldehyde or the oxo group of donors, and this for only the hydroxyl group donors. This aldehyde or the oxo group donors and so on. You can find the subclass like this. واضح يا شباب؟
واضح.
معلش يا بنت، يعني لو في حد ما فاهم ممكن ارجع بالعربي. هل نحتاج نعملها بالعربي؟ طيب الجزء الثاني من المحاضرات هو الجزء الخاص بالميكانيزم اوف اكشن في نظريات كثيره، لكن اهم شيء في الموضوع ده انه نفهم الاكتيف سايت بتاع الانزيم. وبما انه قلنا الماجوريتي اوف enzymes are type of proteins and we know that proteins have three dimensional structure زي ما ذكرنا البارح very specific for each type of protein and identifying the protein type and its function. So the three-dimensional structure of the albumin, for example, is very unique for that albumin, will not be typical to that of hemoglobin, as both, although both are like globular protein, plasma proteins, they work in the plasma, and they are soluble in the plasma aqueous media, and both of them are globular shape، شكلهم عام شكل كروي، لكن السبيسيفيك 3-dimensional structure بتاعهم بيختلف بصورة كاملة، وكل واحد منهم ال 3-dimensional structure بتاعته خاصة به بصورة يونيك جدا. ذكرنا في المحاضرة الماضية تتذكروا انه عشان نصل لل 3-dimensional structure بنمشي عبر مستويات بتاعت بروتين structure، primary structure خاصة بالسكيلتون بتاع الامينو اسيد سيكونس، والسكندري structure الخاصة بالالفا هيليكس والبيتا شيتس، والتيرشري structure الخاصة بالتجمع بتاع الالفا هيليكس والبيتا شيت بروابط غير تساهمية عشان يدونا بداية شكل ثلاثي الابعاد الخاص بكل بوليبتايد تشين. قد يكون البروتين ينتهي في المستوى ده، قد تكون بروتينات ثانيه تمشي لمستوى لغايه المستوى الرابع من تكوين البروتين اللي هو التيرشري كواترنري ستراكشر من البروتين، اللي هو بيكون فيه 2 or more separate polypeptide chains كل واحده منها ماخذه 3 dimensional structure كامله، وبعد ذاك تجي تفصلها مع تعمل بايندينج وبوندينج مع مركب اخر او بوليبتايد تشين اخر. So الانزيمز بتنتمي للجروب ده من البروتينات بصوره عامه، هي higher order of proteins. اغلب الانزيمز تكون من الكواترنري ستراكشر ليفل من البروتين، عندها active site بمعنى انه مكان بيتم فيه ارتباط السبستريت اذا كانت وحده او اكثر، في الغالب تكون سبستريت وحده، والاكتيف سايت ده عنده خصائص وعنده ميزات حنذكره بعد شويه. فاذا تخيل انه الشكل ده اللي انا بشير له هو الانزيم، والشكل العام شكل الاسنان دي ولا الجروفز دي الاكتيف سايت بتاعت الانزيم، بتكون very specific للسبستريت بتاعتها. So the substrate can bind and the enzyme by binding to the substrate by binding to the enzyme it will be transformed slightly, and this type of the binding is known as the enzyme substrate complex. Then some changes will take place in the substrate, making it as like an initial type of product, but it still bind with the enzyme, so it's known as enzyme product complex. And finally, the enzyme will release the product into the final form. It can be two types of product, like the example, the hexokinase, which will add the substrate, which will activate the addition of the phosphate to the glucose, and this is one product, or the acetylcholine esterase, which can split the choline and the ester acetate into two products. So this is general the active site. This active site, if you can observe, it is not typical to the shape where the substrate can bind, and this gives some sort of flexibility of the active site of the enzyme, which can be affected by many factors. And later we'll know in the mechanism of action how these enzymes mechanism have been explained or described by different theories. So one of the theory indicates that the active site of the enzyme is very rigid and should be typical to the binding site of the substrate, so that they can fit and give complete compatibility or complete attachment. Other theories, which is explained by this example, give some sort of flexibility in the active site where it can be slightly modified or change slightly from this not typical curved shape to the typical shape of the substrate, like this case. So you can easily recognize that this one is not typical to this shape because there's some sort of transformation has changed, been made to the active site to fit exactly with the binding site of the enzyme. So we'll go through this in the subsequent slides. So the enzyme kinetics, it is a quantitative measurement of the rate of the enzyme catalyzed reactions and the systemic study of the factors that affect this rate. As we mentioned in the introduction, the enzymes can activate the reaction or accelerate the reaction by a factor of up to 1 to the power 6. That means by millions. Can be 2 types to power 10 to the power 2 by 10 to the power 6. Can be 5 by 10 to the power 6. So 5 times or 5 million and so on. So this study of this rate of reaction and how it will catalyze the rate of reaction, accelerate the rate of reaction, is what was known as enzyme kinetics. A similar type of study and scientific branch of the pharmacology is known as pharmacokinetics, which is specific for the study of the changes that will take place in the biological system to the specific type of pharmaceutical preparation or drugs or medications. From the time of the injection, if we talk about injection, for example, to the absorption, to the metabolism, and to the binding with the specific target substrate where it will do its medication effect, and to the time of the excretion, if there is any residue that will be excreted, or the complete metabolism of the medication or the drug. So the enzyme kinetic is almost similar to that, but we are studying the changes that will take place for the rate of the enzyme catalyzed reaction and any factor that will affect this rate of reaction. For example, the time of the reaction versus the energy needed for any specific reaction is plotted into this example. So if this red circle stands for the substrate, if the substrate is we want to change this substrate to the product in the black spot here, we need an activation energy which is equivalent to the amount from C up to A. This is reaction energy, activation energy, which we need to transform this substrate to the product. And the activation energy, just for reminder, it is the energy need to change the molecules of the specific substance, in this case the substrate, from the ground state to the excited or the activated state. يعني المركبات يا شباب في الكيمستري ما بتدخل في تفاعلات كيميائية ولا في تغيرات وتشنجز ولا تكون عندها قابلية انها تتحد او تنفصل من مركبات اخرى الا اذا يعني تعرضت لمقدار من الطاقة الابتدائية بتخليها تكون excited او يعني فيها شيء من الإثارة والتحفيز من الجراوند ستيت اللي هي فيها تكون نورمال وستلد وستيبل لاكسايتد ستيت بالاكتيفيشن انرجي. So one of the mechanism of the enzymes is to reduce the activation energy from this C to A amount, if you can imagine as the amount of the energy needed for the activation without the enzyme, to the acceleration of the enzyme by reducing this energy to the amount only between C and B. And in both cases, we'll end up with the same product, but with less energy. And this one is not typical, but the product should be reached in the enzyme catalyzed reaction around this point. لأنه يعني reducing the activation energy at the same time accelerating the reaction. So we expect the enzyme catalyze the action to end up with the product at this position, but in this specific example, it's not typical to what we need. This is only concentrating on the reduction of the activation energy which is needed by or achieved by the enzyme catalyzed reaction. Examples, activation energy of sucrose. We can take it as an example in this case. If we want to split the sucrose disaccharide into glucose and fructose by normal acidic hydrolysis, it will need an activation energy which is equal to 26,000 calories per 1 single mole. This is the acid hydrolysis without an enzyme. So the energy needed for the acid hydrolysis of the sucrose into glucose and fructose is equivalent to 26,000 calories for each single mole of sucrose to become glucose and fructose. The same reaction can take place by the sucrase enzyme, which will reduce activation energy from 26,000 in the acid hydrolysis to only 9,000 calories per mole. So this is what we mean it by this reduction of the energy. 26 minus 9,000 calories. This is the difference which can be achieved by this specific example. واضح يا شباب؟
Can you repeat the part
واضح.
acid hydrolysis and sucrase?
نعم تاني.
Can you repeat the part of acid hydrolysis and sucrase hydrolysis?
Okay. So if we take the sucrase or sucrose, sorry, as an example, it can be split into constituents which are the glucose monosaccharide and the fructose monosaccharide by different ways. One of them is acid hydrolysis by adding an acid. And you know from the previous lecture or from your previous knowledge that the addition of any acid will change and will break down the bonds between the polypeptide chains or the polysaccharide chains, the glycoside bond, because of the acid hydrolysis. So to split the sucrose into glucose and fructose by acid hydrolysis by adding an acid only, it will need activation energy equivalent to 26,000 calories by each mole of sucrose. While if we replace the acid and hydrolyze the sucrose by the sucrase enzyme, so the acid hydrolysis has been replaced by the sucrase hydrolysis, and remember that the hydrolase are the class of enzymes that will split the molecule into two by adding the water molecule. So the sucrase will reduce the activation energy from 26,000 in the case of the acid hydrolysis to only 9,000 calories per mole for the splitting of the sucrose into glucose and fructose. واضح يا شباب؟ يعني ممكن نعمل السكروز لمكوناته الأساسية بتاعت الجلوكوز والفركتوز إما بإضافة أحماض، أي حامض عضوي ولا حامض في المعمل نضيف سيتوزام مخفف أو سيتو إصافر مخفف، بيحتاج لأكتيفيشن إنرجي عشان يحصل الجلوكوز والفركتوز ينفصلوا من بعض بشكل السكروز بالأسيد هيدروليز لطاقة تعادل 26,000 كالوريز لكل مول من السكروز. في حين السكريز انزيم بيقلل الطاقة دي إلى only يحتاج لطاقة تساوي 9,000 كالوريز، بمعنى إنه يقلل الانرجي نيدد بمعدل 14,000 15,000 17,000 كالوري لكل مول. فده الريدكشن اوف ذا اكتيفيشن انرجي، وبالتالي حنصل للاند برودكت بصوره اسرع مما لو كانت في حاله الهيدروليز ولا اي نوع من الهيدروليز ثاني. الميكانيزم اوف اكشن اذا المثال السابق ده وضح، في عندنا مجموعه من النظريات، واحده منها بتقول انه الانزيم سبستريت اكشن بياخد الفورم ده الاي لانزيم الاس للسبستريت والاي اس للانزيم سبستريت كومبلكس. Whenever you have an enzyme and substrate, you add the substrate to an enzyme, they will form the enzyme substrate complex as intermediate molecule. يعني أي إضافة أي substrate لإنزيم أو إنزيم لسبستريت اعتبرنا إنه السبستريت هي constant والإنزيم هو changing. عندنا بيكر حطينا فيه سبستريت معينة بس assume إنها هي جلوكوز في محلول بتاع ووتر، أضفنا له هيكسوكاينيز اللي هو الإنزيم بيدينا في البداية انزيم سبستريت كومبلكس ES. الخطوة الثانية تكون هي الانزيم سبستريت تو برودكت، يحصل ترانسفورميشن من الانزيم سبستريت كومبلكس في خطوة انترميديت لانزيم برودكت كومبلكس، بعد ذاك يدينا انزيم بلس برودكت. البرودكت هي في حالة الجلوكوز والهيكسوكاينيز اللي هي جلوكوز 6 فوسفيت إذا اعتبرناه كمثال. ممكن ننظر للرياكشن ده as overall باعتباره enzyme plus substrate will give us enzyme substrate complex. So some changes will take place in the substrate to form the product, and some theories they put the enzyme product as one of the intermediate also product of this reaction, and at the end the enzyme will come up splitted from the product without being changing or altered or with having any consumption or being used, and the substrate will be transformed into the product shape. إذا أخذنا كلام ده يا إخواننا بالمثال بتاع الجلوكوز والهيكسوكاينيز، قلنا الإنزيم هو الهيكسوكاينيز والسبستريت هي جلوكوز، يدونا انزيم زائد سبستريت كومبلكس كمركب يعني كومبلكس في البداية، وممكن يدينا انزيم برودكت كانترميديت موليكيول أو برودكت، بعد ذاك يدينا انزيم زائد برودكت. الرياكشن بياخد المسار ده. فالميكانيزم اوف اكشن بتاع ارتباط الاثنين ديل عندنا two different ways. enzyme increase activation or the action rate by decrease activation energy as we mentioned, while the enzyme substrate interaction can be explained by the following example. ما أعرف شايفين الصورة دي كلها ولا لا، لكن هنا ده example للسبستريت الانزيم والشكل بتاع التو hexagonal و pentagonal shape complex دي السبستريت يدونا انزيم substrate complex، وبعد ذاك الانزيم will act and will give enzyme changing slightly in the substrate by splitting this bond for example, and it will give the enzyme product complex which is not mentioned in these stages and I mentioned it verbally. At the end we'll end up with the enzyme plus the product, which is in this case two separate units. So we can imagine this example typical to the acetylcholine. This is acetylcholine. We can imagine this as acetylcholine. The substrate acetylcholine and the enzyme acetylcholine esterase. Acetylcholine esterase plus the acetylcholine will give us the complex of acetylcholine esterase and the acetylcholine, and then the transformation by splitting the ester bond will take place, but it still will give this complex with the acetylcholine esterase enzyme and the choline and the acetate unit, and then this will be intermediate step like this one, and finally we'll end up with the acetylcholine esterase as it is without being changed or altered, and the product will be the choline and the acetate unit. There are two theories explaining the mechanism. One is known as the lock and key model, and the other one is known as the induced fit model, and how the enzyme will bind to the substrate and will do its transformation and changes into the substrate to form it into the new product. I've got two theories. The first one is the lock and key model, which stands or states that the active site of the enzyme has a rigid shape, just typical to the shape of the binding site of the substrate. So only that specific substrate will fit with the shape of the active site of the enzyme, and this will play a very good role in explaining the enzyme specificity and to some extent the enzyme selectivity. By that, each enzyme is just specific to its substrate, where its active site cannot fit any other type of substrate. But this theory or model of explaining the enzyme mechanism is explained by this image. This is the key, and this is the lock. The key, and I have got specific type of teeth like, and there is some grooves in the lock which will fit typically to that specific. And this, you know, this is very rigid, and this explains, as we said, the specificity of the enzyme, and explains the loss of the activity if the enzyme active site has slightly changed. It will lose its activation property. It will be like the deactivated enzyme or protein. This is the example of an enzyme which explains the lock and key model. This is the active site with this V shape of the enzyme, and the substrate has a shape where some of it is structure can fit typically with this site. So this will explain exactly how rigid this shape is and how it will fit exactly to this specific substrate. If we change it by any other substrate without having this specific binding site, it will not fit. So this theory can explain some of the mechanism of some of reactions of the enzymes, but not all type of enzymes, because we have got some like a remarkable number of enzymes that have got some sort of flexibility in the active site, which can be slightly modified to fit with the substrate, and this will be explained by the induced fit model, which has been stated by the Koshland's theory. The induced fit model states that the active site of the enzyme has some sort of flexibility. It's not typical rigid, so it can re-modify its shape to fit with the substrate to adjust itself. And this is an example. We have this enzyme with this active site having this specific shape, and our substrate have this type of binding site. So it is not typical to the active site of the enzyme, but the enzyme have some sort of flexibility that can rearrange itself in the active site to fit exactly with the substrate and to make the changes which are expected to be done by this enzyme, and this is known as the induced fit theory. And in this case, the substrate or the enzyme has the flexibility of adjusting itself or the active site to fit with the maximum possible binding forces. Remember, the forces which would bind any substrate with its enzyme active site are usually non-covalent bonding. There is no any covalent bond between the enzyme and the substrate, whether in the lock and key theory or the induced fit theory. All the forces which will bind the substrate to the enzyme, whether in this case or the previous case, are non-covalent bonding. So this type of the enzymes have some flexibility that can be modified to fit with the substrate. The greater range of the substrate specificity, that you can find this type of the enzymes with that flexibility, they can be of broad spectrum rather than just rigid to very specific enzyme or very specific substrate. This model is known as the wider range of enzymes will match this, will be explained by this theory, and we'll see some examples in the subsequent slides. واضح يا شباب؟ حتى إنه المحاضرة حطوه شوية. أظن الآن وأتوقع إن إحنا في نصف الطريق، أو إذا مشينا بربع ساعة ممكن نخلص في نص ساعة إن شاء الله. تكون زي البارح. So the factors that affect the enzyme activity and the mechanism of the enzyme by reducing the rate of reaction or accelerating the rate of reaction. This is how we study the rate of reaction by second. So one time taken for changing the substrate into the product. There are so many factors that can affect the enzyme activity and the mechanism of enzyme in doing its activation. One of them is the concentration of enzyme. The number two is the substrate concentration, the product concentration, the temperature of the reaction medium, and the hydrogen ion concentration or the pH of the media, and the presence of activators or the presence of some inhibitors in some cases, or the presence of repressors or depressors, or the covalent modification which can take place due to any conditions, whether intentionally needed or unintentionally present as a situation of the action. So we'll take example of these different factors. The first one is the enzyme concentration and how it will affect the rate of the reaction. As you increase, in this example, as you increase the amount of the concentration of the enzyme, the velocity of the reaction or the speed of the reaction will be increased proportionally, giving this type of proportional relationship, directly proportional. The rate of reaction or the acceleration of the reaction is directly proportional to the enzyme concentration. In other words, if we keep specific amount of the substrate constant, let's say 10 millimoles of the substrate of the glucose, and we add the hexokinase in different amounts and different concentrations, starting by example 1 millimole, 5 millimole, 10 millimole, 15 millimole, and 20 millimole. We'll find the rate of the reaction will increase by increasing the concentration of the reaction where we put the substrate as constant. This is the first factor. So the relation between the enzyme concentration and the velocity of the reaction is directly proportional. As we increase the enzyme concentration, the velocity or the speed of the reaction will be increased proportionally, giving this type of the relation. Regarding the substrate concentration, this yellow shapes, the C-type shape, are like examples of an enzyme, and we kept it in this example as constant, and we changed the concentration of the substrate, which is the red circle or the red shape. So if we start by giving a low concentration of the substrate to the same amount of the enzyme, we expect that the rate of reaction will be very slow, because only one of the substrate will bind to one molecule of the enzyme. And let's say instead of talking about millimoles, let's talk about molecules. We have 6 molecules of enzyme, and in the starting we start with only 2 molecules of the substrate. So the rate of reaction will be, when we add only 2 molecules of the substrate, will be very low at this level. If we add 6 moles of the substrate, the chance of the binding of the substrate with the enzyme will be increased by slightly increase, and will reach up to 50% of the substrate have been attached or bind to 6% to 50% of the enzymes, and this is known as V50. The speed of the reaction will be at 50% of the maximum possible reaction or the rate of reaction, where we have 50% of the enzymes have been binding to the present substrate. If we increase the amount of the substrate, we expect some extra binding, so the rate of reaction will be increased slightly, and with not very direct proportionality, but as we increase the rate of the reaction, the concentration of the substrate, we will find that the rate of reaction will increase gradually, not proportionally, but gradually it will increase, till it reaches a point where we call it the maximum velocity of the enzyme activated reaction, where all the substrate has been attached or bind to the enzyme, and this is the maximum possible velocity that can be reached. After this, no matter how much we add from the substrate to the same amount of enzyme, it will not change the speed of the reaction. So this is the maximum velocity that can be reached, and it's abbreviated by Vmax. So I make this animation to make it easy for you to follow. At the starting, where we add only small amount of the substrate, the rate of reaction was very slow, around this range only. When we add more substrate and we reach the 50% velocity, this one 50% velocity, and this will be at the km of the substrate, or the amount of the substrate. 50% will bind to the enzyme molecules. So this is known as the km, the rate at which we reach 50% of the velocity. Then, if we take the example of the hexokinase and glucokinase, both enzymes work on the glucose by adding the phosphate group or transferring the phosphate group from the adenosine triphosphate to the glucose unit. The hexokinase, you may know that it is one of the enzymes, the transferases, that work in all cells except the liver. It has got a very high binding capacity with the phosphate group, and it is insulin dependent enzyme. So it can reach the km, or the velocity of 50% attachment of the hexose sugar, the glucose in this case, with the phosphate group at a concentration of 0.05 millimole per liter. While the isozyme of the same nature or same activity, the glucokinase, is specific for the liver, and it is insulin independent, but it can reach the Vmax of the attachment of the hexokinase enzyme with the substrate with the glucose to have the transformation of glucose to glucose 6-phosphate at V50 only on when we reach a concentration of 10 millimole per liter. So this means we need more enzyme of this, more of this glucokinase than the hexokinase, which is insulin dependent enzyme. When we go further to the maximum velocity which can be reached, where we have all the enzymes bind to all the possible substrates, no matter if we need, or if we add more substrate, for example, in this case glucose, it will not affect the rate of the reaction. Will go just like plateau line without changing. This is a V maximum which can be reached by this type of reaction. واضح يا شباب؟ واضح؟ في سؤال؟
واضح.
طيب جميل. نمشي للسلايد اللي بعديها اللي هي الفاكتورز افكتنج، من ضمنها غير الانزيم كونسنتريشن والسبستريت كونسنتريشن عندنا التمبريتشر اوف ذا اكشن. معروف انه التفاعلات الحيويه في الجسم كلها بتتم في الفسيولوجيكال تمبريتشر بتاعت البودي، هي around 37 degrees centigrade، ودي الموست اوبتيمم تمبريتشر بيحصل فيها اغلب التفاعلات بتاعت الرياكشن بتاعت الديفرنت تايب اوف انزيمز. فاذا تخيلنا اي واحد من الانزيمز وده نشوف الفيلوسيتي بتاعت الرياكشن، اذا بدينا بتمبريتشر slightly less than 37، let's say around this 30 degrees centigrade، الحته دي على البوينتر لو شايفينه، the rate of reaction will be less than the maximum possible velocity of reaction, because it's not the optimum temperature. So as we increase the temperature of the in the medium of the reaction, the rate of reaction will be increased till we reach the optimum possible temperature. الكيرف ده يا شباب عاملينه في شكل بتاع bell shape عشان يوضح a normal distribution between the rate of increasing the rate of reaction with the increasing temperature till we reach the optimum temperature, and how it will be affected inversely affected by the increase of extra heat or heating of the medium by going back down to as low as almost zero reaction. But الشكل ده يا شباب ما هو التيبيكال اللي يحصل في الجسم بتاع الانسان. هنا بس عايزين نوضح انه العمليه بتاعت التناسب معدل التفاعل او سرعه التفاعل مع التمبريتشر بتاعت الميديوم اللي بيحصل فيه السبستريت والانزيم اكتيفيشن ستيت بتبدا من لو ريت في لو تمبريتشر اللي هي اقل من الاوبتيمم بمستوى معين. ممكن ناخد الجزء الاعلى من البيل شيب ده لغايه ما نصل للماكسيموم بوسيبل rate of reaction which can be at the optimum temperature. طيب ليه عملنا المدى ده؟ لانه بعض الانزيمات بتعمل في مستوى طاقه مستوى في درجه حراره slightly less than 37، ممكن تكون لغايه 36 35 point something، وممكن تكون لغايه 37 point something. Still enzymes can be activated or active in this rate, and around this will be the optimum temperature of each specific enzyme. So التمبريتشر بتأثر في التفاعل. As we increase the temperature, the rate of reaction will increase slightly till we reach the optimum temperature. And we don't expect that we can start a reaction at a temperature of 10 degrees centigrade and starting increasing the temperature with for the same enzyme substrate reaction till we reach, for example, 30 degrees. ببساطة يا شباب لأنه نتوقع إنه النوع ده من change of the temperature will affect the nature of the three-dimensional structure of the protein of the enzyme. It will lead to denaturation either by increasing that 20 degrees or by, if we take it the opposite way, by decreasing the temperature up to 10 instead of 30 or 37. عاوز أقول بمعنى آخر يا شباب الأوبتيمم temperature بتاعت الرياكشن عادة between 35.5 for example up to 37.5 أو to 38.5 for example. قل له إنزيم عنده optimum temperature بيصلها في مستوى معين. الشيب ده ما تيبيكال اللي بيحصل في الأكشوال living cells، لأنه ما في رياكشن بيتم في حرارة 10 ولا 20 ولا حتى 25. ممكن في الروم تمبريتشر الخارجيه يكون ده المستوى بتاع الرياكشن التمبريتشر كده، لكن في داخل الجسم كلها تكون around 37 plus or minus slightly. فالفكره الاساسيه انه اذا زدنا التمبريتشر after the optimum temperature the reaction will go down and it will be slow till we lose all the reaction or the velocity of the reaction will be zero simply by denaturation. The other example is the pH. The effect of pH of the medium of reaction again will be like this bell shape. As we increase the pH of the reaction for a specific enzyme, the rate of reaction will be increased as we increase the temperature, the pH, till we reach the optimum pH of that specific enzyme. And we have used three examples in this case or this diagram. The first one for enzymes that activate or they do their function in the acidic mediums like the gastric enzymes, as you've seen as an example. It will have its optimum pH at pH equal to 2. Okay? So if we start by a pH of 1.5, somewhere around this point, we expect the rate of reaction of the pepsine for a specific substrate, whether protein or lipid, to be slightly low. And as we increase the pH, the rate of reaction will be increased till we reach optimum pH, which is equivalent to, in this case, almost 2. So this is the optimum temperature of the optimum pH of the pepsine. And if we increase the pH more to 2.2, for example, 2.5 to 3, the rate of reaction will go down and will be slow till we lose all the reaction at pH equal to 4. For the amylase, which we have usually in slightly alkaline media like the mouse or the intestine media, which is the epsilon will be around 7 point something, almost closer to 8. And if we increase the rate of the pH of the reaction for the amylase from 6.5, for example, we will have a low rate of reaction, and as we increase the pH, we will increase the rate of reaction till we reach the optimum reaction at the pH, which is 7 point, for example, 5 or 7 point something, very close to 8. For the arginase enzyme, it will work in a typical alkaline medium, and again the rate of reaction will be increased gradually until we reach the optimum pH, and if we increase the pH anymore, it will go down. So there is some similarity of the effect of the temperature and the pH, but here we have got different type of enzymes with different medium of reaction, acidic, semi-alkaline, or typical alkaline media, and each will have an optimum pH where it will give the maximum possible velocity. Any increase of the temperature or the pH or a decrease will go to reduce the rate of reaction by the proportional way. So the effect of these factors again can be summarized into this diagram. The pH will have a normal proportionality in the specific medium of the enzyme activation, whether acidic, semi-alkaline, or typical alkaline media, until we reach the optimum pH. And regarding the temperature of the reaction will increase by increasing the temperature till we reach the optimum, and then will decrease by any extra heating. And the substrate concentration is completely different because it will go gradually increasing by increasing the concentration of the substrate till we reach the V maximum at the maximum binding possibility of the enzyme and substrate. Any addition of extra substrate will not affect the enzyme catalyzed reaction by any change, because there is no more enzymes to be bind. And this can be explained by this example. Here we have got only 6 molecules of the enzyme, and the binding only at the starting A, only one enzyme and substrate have been bind to form the rate of reaction, which is very low. And now in this case we have got medium, or we can say moderate binding between the substrate and some of the enzymes. Substrates have been not completely bind. This we can imagine as intermediate reaction rate. And the time wave, when you have the maximum binding in this case, no matter how much we add from the substrate concentration, it will not affect the rate of reaction, because all the enzymes have been bind to the subsequent substrate, so it will not increase anymore the rate of reaction after the Vmax of the substrate concentration which has been reached. Okay. The inhibitors, the last part of the lecture. The inhibitors are substances that will affect the rate of reaction by preventing by preventing the enzyme substrate complex, or by preventing the production of the specific or selected product. So the enzyme inhibitors prevent the formation of enzyme substrate complex, and they will not give us the end product which we expect, either in the amount or in the nature itself. We have got different type of inhibitors. They are classified generally into 2 types: either reversible inhibitors, which can be where the reaction is affected by the inhibitor, and as if we remove the inhibitor, the rate of reaction will be reversed back to the normal. And some type of inhibitors which are known as irreversible inhibitors. So the inhibitors of enzymes are classified into 2 major classes: reversible inhibitors or irreversible inhibitors, according to their nature. If they have been added to substrate enzyme reaction, they will inhibit the reaction. If they have been removed, they can go back to the normal state. But this type of the inhibitors is irreversible, or it will not go back to the normal. So the enzyme inhibitors can be summarized in this diagram. They can be classified into reversible inhibitors or irreversible inhibitors. The reversible inhibitors can be of 3 different types or different classes. Some of these reversible inhibitors are known as competitive inhibitors that compete with the substrate with the active site of the enzyme, or some are known as uncompetitive inhibitors, and the last class is known as the non-competitive inhibitors. I will go through some examples for each of these in the subsequent slides. أي سؤال يا شباب so far؟ المثبطات بتاعت الانزيمات يا شباب هي مواد تمنع إما التحام الانزيم والسبستريت عشان يدونا الترانسفورميشن المتوقع من الرياكشن، أو تعمل changing في الانزيم active site بحيث إنه تمنع الارتباط، أو يكون في يعني تأثير آخر غير قابل لأنه يعني يرجع للشكل الطبيعي. بتتقسم بصورة عامة لمثبطات عكسية ومثبطات غير عكسية. مثبطات عكسية بالعربي بتتقسم لمثبطات تنافسية ومثبطات غير تنافسية ومثبطات بيسموها بالعربي لا تنافسية. إذا صحت الترجمة دي، لكن المفهوم المقصود هو في من الإكزامبل الجاية اللي هنشوفها في السلايدات الجاية. الكومبيتيتيف انهبيترز واحدة من كلاس بتاع الريفرسبل انهبيترز اللي هي مثبطات تنافسية. دي مركبات هي عبارة عن سبستريت انالوج تشبه السبستريت في شكلها الكيميائي وفي طبيعتها وفي الاستراكشر بتاعتها، او اتليست في البايندينج سايت مع الاكتيف سايت بتاع الانزيم. So it will compete with the enzyme with the substrate in binding with the active site of the enzyme. ففكرتها انها يعني مركبات تشبه السبستريت في تركيبها كيميائي وفي شكلها اتليست في الجانب بتاع الاكتيف سايت بتاع البايندينج، وبتنافس السبستريت في الارتباط مع الاكتيف سايت بتاع الانزيم، وده مثال لها في النموذج الجاي. تأثيرها بيكون انه تأثير تنافسي، وهنا يمكن الدياجرام ده بيوضح. عندنا ده انزيم معين، وده الفي شيب بتاعه ده الاكتيف سايت بتاع الانزيم، والجزء العامل بشكل يعني كيرفد جروف من الانزيم ده بيسموه بروسيتيك سايت. ممكن تطبقوا مركبات اخرى ده بروسيتيك جروفز اذا كان نون بروتين موليكيولز ولا غيرها، لكن اللي بيهمنا في المثال ده الاكتيف سايت بتاع الانزيم ده عنده دي السبستريت بتاعته الطبيعيه. اذا اعتبرنا ده الهكسوكاينيز وده الجلوكوز، الاكتيف سايت بتاع الهكسوكاينيز عنده تطابق او تشابه مع البايندينج سايت بتاع السبستريت. So the substrate will bind to give the substrate enzyme complex في الأحوال العادية. الانهبيتر اللي هو competitive و reversible بيشبه السبستريت في at least في this binding site، ما بالضرورة تبقى في كل الشيب، عشان كده نسميه السبستريت انالوج. والانالوجز يا اخوانا يعني هي مشابهات المركبات ومشابهات المواد. فلأنه عنده نفس الشيب بتاع البايندينج سايت بينافس السبستريت في الارتباط بالاكتيف سايت، وبيمنع او بيحول دون ارتباط السبستريت مع الانزيم، وبالتالي بيعمل بروهيبيشن للانزيم سبستريت كومبلكس، وبالتالي ما حيكون في برودكت. الكلام ده يا اخوانا اعتبرنا عندنا 1 to 1 يعني 1 انزيم موليكيول و1 سبستريت موليكيول 1 انهبيتور موليكيول. الانهبيتور موليكيول ده كومبيتيتيف بيتنافس مع السبستريت وياخذ مكانه بدل الارتباط مع الانزيم اكتيف سايت بالضبط الانالوج او الانهبيتور. اذا اعتبرنا عندنا يعني different concentration for example 10 مليمول من الانزيم او 10 موليكيول من الانزيم و10 موليكيول من السبستريت و10 موليكيول من الانهبيتور النوع ده من الكومبيتيتيف، وهذناهم كلهم في ميديم واحده ضفناهم في let's say في بيكر ولا كونيكال فلاسك. بوث السبستريت والكومبيتيتيف انهبيتور بتوقع انه يكون في بعض الانهبيتورز تتحد مع بعض الانزيمز بمعدلات ممكن تصل ل 30% 40% 50% او 70%. It depends على البايندينج كاباسيتي بين الاكتيف سايت بتاع الانالوج مع الاكتيف سايت بتاع الانزيم. So at least we'll prevent from getting up the expected amount of the enzyme substrate. إذا عندنا 10 مليمول أو 10 molecules من الانزيم و 10 molecules من السبستريت، في النورمال يفرض لنا 10 products أو 10 مول من البرودكتس. في وجود الانزيم الانهبيتر ده الكومبيتيتيف، متوقع انه كميه البرودكت هنا ما بالضروره تكون كومبليتلي نو برودكت، ممكن تكون في برودكت لكن بتقل بمعدل ارتباط السبستريت مع الانزيم. فاذا كنا قلنا ارتباط السبستريت مع الانزيم ب 50% بيكون البرودكت تنقص ب 50%، بمعنى انه 50% اونلي من الانزيم يكون عندها قابليه ترتبط مع الانزيم، لانه ال 50 الثانيه ترتبط مع الانالوج وان سو اون. واضح يا شباب؟ النوع الثاني من الانزيم انهبيترز اللي هو النون كومبيتيتيف، اللي ده نوع من السبستريت او المثبطات او المواد اللي بتعمل على يعني تثبيط التفاعلات الانزيميه بالارتباط بمكان اخر، ما هي انالوج للانزيم ولا بتنافس في الاكتيف سايت، لكن ترتبط مع جزء اخر من الانزيم زي البروسيتيك جروف اللي ذكرناه قبل شويه في المثال، وبالتالي تغير من الاكتيف سايت بتاع الانزيم وتمنع ارتباط السبستريت مع الانزيم. يعني هي ما تنافس الانزيم في ارتباط مع الاكتيف سايت، لكن تنافسه او ترتبط مع مكان اخر تغير الاكتيف شيب. سو اذا تخيلنا ده الانزيم بتاعنا اجين الاكسوكاينيز، وده الجلوكوز بتاعنا، وعندنا سبستريت او سوري انهبيتر سبستريت ماده اخرى ما عندها علاقه بشكل السبستريت بتاعتنا الجلوكوز، قد تكون any type of any other type of groups، لكن عندها similarity في البايندينج سايت مع البروسيتيك جروف هنا اللي ممكن يرتبط مع الانزيم. ارتباطها هنا لانه الانزيم ده قد يكون فيه دايما فلكسبيليتي، بيتغير الاكتيف شيب ده من شكله الطبيعي بتاع الارتباط مع الجلوكوز للشكل ده. بالتالي ما حيكون في فرصه لارتباط السبستريت مع الانزيم. فهنا ارتباط بتاع النون كومبيتيتيف انهبيترز بالديفرنت position او different site of the enzyme اللي هي protetic site، و changing the active site of the enzyme and preventing the binding of enzymes as substrate complex, so it will give no reaction between the one-to-one substrate enzyme and one inhibitor in this case. Again, if we assume we have got 10 moles of the enzyme, 10 moles of this substrate, and 10 moles of this inhibitor, we may expect that only some of these inhibitors can bind to the groove of the non-prosthetic site, and the remaining will be not affected. So if we expect that we have 30% of the inhibitor in this case bind to the enzyme, only 30% of the enzyme will be changing in the active site, and the remaining 70% can bind with the enzyme and give the 70% product, and so on. بمعنى آخر بمعدل ارتباط النون كومبيتيتيف انهبيتور مع هذا سايد من الانزيم وتشينجينج الاكتيف سايد، بيحصل التغيير في الرياكشن او معدل الريدكشن في الرياكشن ريت والبرودكت المتوقع بالنسبه للنوع ده من التفاعل. الان كومبيتيتيف انهبيتورز ودول كلهم يا شباب الكومبيتيتيف انهبيتور والنون كومبيتيتيف انهبيتور، والجاي الان الان كومبيتيتيف انهبيتورز كلهم من الكلاس الاول بتاع ريفرسبل انهبيتورز. النوع ده من الان كومبيتيتيف انهبيتورز هي لا مواد عندها علاقه بالارتباط مع البروسيتيك جروب ولا الاكتيف سايت، فما عندها تنافسيه في الاكتيف سايت ولا عندها ارتباط مع البروسيتيك جروب، لا هي عندها فرصه الارتباط مع الانزيم سبستريت كومبلكس. بعد ما يتحد الانزيم والسبستريت بالطبيعه بالصوره الطبيعيه بتاعتهم يدونا انزيم سبستريت كومبلكس. الانهبيتور ده من النوع بتاع الان كومبيتيتيف يرتبط مع الانزيم سبستريت كومبلكس، وبالتالي ما حيخلي الانزيم يعمل اكسبكتد تشينجز في السبستريت ويدينا برودكت منها تو بي ريليز از برودكت. فهنا الارتباط بتاع الان كومبيتيتيف انهبيتورز هو مع الانزيم سبستريت كومبلكس and preventing the transformation changes that can be expected for the substrate and preventing the release of the transformed product substrate into the product. يعني مركبات الانكومبيتيتيف انهبيتورز دي مثبطات ترتبط مع الانزيم والسبستريت كومبلكس، وبالتالي تمنع تغيير الانزيم السبستريت عشان تدينا البروتين المتوقعه، وتمنع releasing of this substrate which have been transformed. So any one of these three types of the inhibitors, if we remove the inhibitor, the reaction rate will go normally without affecting, and this is why we call it reversible inhibitors. يعني كان عندنا inhibitor ضفناه هنا عمل التأثير ده، إذا أزلنا أو إذا زدنا السبستريت concentration الرياكشن ريت بيمشي طبيعي والرياكشن يكون reversible. أما إذا مشينا للكلاس الأخير اللي هو ال irreversible inhibitors، دي مركبات بترتبط مع الإنزيم وبتعمل فيه chemical change في النشر تبعه، إما forming new covalent bonds أو breaking present covalent bonds. So changing the three-dimensional structure of the enzyme as whole, or changing the active site of the enzyme by forming a new covalent bond or changing the covalent bonds. مثال لها المواد ممكن تأثر بالنوع ده من التأثير. ادينا التريتنج فاكتور كلها زي الاسيدز والبيزز، تغير البي اتش بتاعت الميديوم اوف رياكشن and you can change the different bonds. and the heavy metals زي الليد والكوبر والايرون، والtemperature increase or decrease. Any of these factors can lead to denaturation by breaking covalent bonds in the enzyme, either at the active site or in other part of the enzyme, or changing the whole nature of the enzyme from the normal three-dimensional structure to be a denatured protein enzyme and to lose its specificity for being binding to the substrate. مثال لها ده folded protein في ال three-dimensional structure بتاعته الكاملة. إذا اعتبرنا ده انزيم معين من انزيماتنا احنا بندرس فيها، وليس اسيوم انه ده الاكتيف سايت بيتحصل فيه ارتباط مع السبستريت. الفاكتورز دي اي واحد منها ممكن يعمل نيو كيميكال بوندز هنا او تو بريك سم كيميكال بوندز وتشينج الثري دايمنشنال ستراكشر بتاع البروتين ده للشكل ده اللي هو انفولدد بروتين، اما في كل اجزائه او اونلي في الاكتيف سايت. لما كان اكتيف سايت في الشكل ده اذا كان التاثير خفيف ممكن بس يفتح الناحيه دي زياده، يكون الذراع ده ماشي في زاويه منفرجه كده. فالتاثير ده تاثير بتاع واحده من الفاكتورز ديل غير طبيعه اما الاكتيف سايت اونلي او غير طبيعه البروتين كله من النورمال بروتين نيتشر اوف 3 دايمنشنال ستراكشر بالاكتيف سايت جروف او active site socket for example a denaturated nature of the protein which loses its specificity for the substrate. The clinical significance of the enzyme في clinical use, many of the enzymes are being investigated or measured for different purposes like diagnosis of diseases, prognosis of a specific type of metabolic disorder or clinical cases, or response to the treatment or follow-up of any medication, how the patient is responding to the medication, and so on. So examples of these enzymes are this table. The amino transferase group of enzyme are used as diagnostic tools for example, myocardial infarction or viral hepatitis. And you can go to any of these examples, and I don't want to spend time because المحاضرة طول الشديد والناس ممكن بعد تمل. أخذنا ساعة ونص exactly. عاوز أحاول أخلص. فالتيبل ده يا شباب ممكن ترجعوا له كل واحد منكم لحاله، وفيه فقط هنا النوع بتاع الإنزيمات وأنواع الدياجنوستيك تيست اللي بيستخدم فيها الفحوصات الإنزيمات دي، وبتدينا indicator لنوع من الأمراض والديس اوردرز. فدي واحدة من الكلينيكال significance of the enzymes in general. وفي clinical significance للإنزيم inhibitors بإنه في الفارماكولوجي بيتم دراسة الديس اوردرز بتاعت الإنسان. إذا أخذنا for example الأنتيبيوتكس as example، كثير من الانتيبيوتكس دي بتعمل في الميكروبيال باثوجينز زي البكتيريا او الفيروسز او الباراسايتس by one of these ways, either destroying or impairing the growth or the invasiveness or the development of the invading pathogens, or stimulating the endogenous defense mechanism of the immune system, or by halting or impeding the apparent molecular process triggered by the genetic, environmental, or biological stimuli with minimal perturbation of the host normal cellular functions. فيعني او بمعنى اخر بالعربي يا شباب الصناعه بتاعت الادويه بيتم فيها دراسه الباثوجينز اذا اخذناها كمثال في الحاله دي عشان نعمل انتيبيوتيك معين لابد ان نعرف الباثوجين ده بيعمل شنو في الجسم. هل يعني impairing الجروس بتاع النورمال جروس بتاع الهيومن او البودي، او بيعمل انترفنشن بانفيزيف over reaction، او developing invading pathogens بيكون في تدخلات من الباثوجين في الباثويز بتاعت الرياكشنز، او الفارماسيوتيك بريبريشن تعمل على انه يعني تحاول تشابه النورمال انتيبيوتيك او انتي بودي ديفنس سيستم بتاع الجسم بتاع الهيومن سيستم، وننتج مركبات تعمل نفس الاميونوجلوبولين فانكشن في محاربه البكتيريا او الفيروس اند سو. فامثله للكلام ده في السلايد الجايه قد تكون اكثر وضوحا. الفيزيوستجمين versus نيوروستجمين inhibitors. الاثنين ديل type of inhibitors يعني مستحضرات صيدلانيه تحضر في المختبر عشان تعمل رياكشن معين او تاكتيفيت رياكشن معين. الاثنين ديل الفيزيوستجمين والنيوروستجمين two types of pharmaceutical inhibitors للستايل sterase enzyme. So this is the enzyme which is targeted by this type of the medication اللي هو physiostigmine, and it will bind reversibly with the acetylcholinesterase and to produce a butyrylcholinesterase enzyme and the acetylcholine. So we need to have the choline and the acetylcholine, the state groups from this reaction. Because the physiostigmine bind reversibly by non-covalent bonds with the acetylcholinesterase to have the acetylcholine, it is known as reversible inhibitor. So الكلمة يا شباب بتاعت typical translation of their inhibitors اللي هي المثبطات قد تدي الامبريشن بتاع انه دائما هي اشياء نيجاتيف. عندنا انزيم معين بيعمل اكتيفيشن لبروسيس، كون يكون في اي ماده تثبط تفاعله او تثبط الدور بتاعه معناها دي حاجه نيجاتيف، لكن ممكن تستخدم بصوره ايجابيه كاز انهبيترز اذا كنا عاوزين نثبط بها تفاعل over reacting or over يعني taking in the specific medium or specific tissue. example لا acetylcholinesterase enzyme ممكن يكون very active في some type of tissues و some conditions أو some disorders، فمحتاجين لأننا نثبت المعدل بتاع التفاعل ده ونخليه يعني أقل من النورمال، فندخل له مادة مثبطة اللي هي الفيزيوستجمين بتتفاعل معه أو ترتبط معه بصورة irreversibly، وهنا نقصد بها irreversibly بصورة بروابط غير تساهمية وتدينا يعني قابلية إنها يحصل لها يعني changing بالremoval. The other medication هو neostigmine bind to the acetylcholinesterase by irreversible binding أو to inactivate the acetylcholinesterase، ولأنه البايندنج هنا irreversible by covalent bond بينشئ رابطة تساهمية جديدة بين النيوستجمين والacetylcholinesterase بيغير الطبيعة بتاعت الاكتيف سايت بتاع الacetylcholinesterase من السبستريت بتاعته، بالتالي يمنع او يقلل من تفاعله وهنا بسميه بالirreversible inhibitor مثبط غير عكسي، بمعنى انه اذا ازلنا النيوستجمين ما حيتغير الريت اوف رياكشن لانه عمل كوفيلنت بونز وغير من التركيب بتاعت الستايكولين ستريز انزيم. Another example الالكترون ترانسبورت تشين فيها كم محطات، واحده من الاكثر الباثويز المعقده في الجسم، لكن عاوز تركزوا فقط في الاسهم اللي فيها نهايتها في علامه الماينس. المالونيت هو انهبيتور للرياكشن بتاع السكسينايد في الكومبلكس نمبر 2 في الالكترون ترانسبورت تشين. الكربون تترا تترا اسمه ايه هو فاتي اسيد، ترايتولوين فاتي اسيد، بيعمل برضه انهبيشن للكومبلكس نمبر 2 بالرياكشن بتاعه، ما يخليه يمشي للسبيس الثانيه. ممكن نختصر الكلام ده بانه از اكزامبل المالونيت كومبيتيتيف انهبيتور للسكسينات في الالكترون ترانسبورت تشين. و2,4 داينيتروفينول again inhibitor للترانسفورميشن of the adenosine diphosphate الى adenosine triphosphate. الرياكشن الطبيعي المفروض يمشي في adenosine diphosphate، وجود inorganic phosphate يدينا adenosine triphosphate. فال 2,4 داينيتروفينول هو inhibitor للرياكشن ده، ودي inhibitor كذلك للتو رياكشنز هنا، واي واحد من ديل انهبيتر لاي من الرياكشنز دي. فالمسج المطلوبه هنا انه في تفاعلات عاديه في الجسم نورمال، يعني رياكشن تحصل في الجسم في الحاله دي في الميتوكوندريا بتاعت اي خليه، يعني في رياكشن بتاعت الالكترون ترانسبورت تشين ممكن تكون في مواد اخرى من داخل الجسم نفسه تعمل على تثبيط التفاعلات في ستيجز معينه، بتكون كومبيتيتيف بانها ترتبط بصوره لا تساهميه، او تكون كومبيتيتيف انهبيتورز بصوره تساهميه وبتغير من طبيعه التفاعل. الانزيمز some of the enzymes can be used as يعني targeting anti-cancer agents like this example. ال 3 برومو بايروفيت كومباوند بيصنع في المختبر عشان يعمل انهبيشن للهكسوكاينيز 2 انزيم، وبيستخدم في التارجتنج الكانسر سيلز. و2 دي اوكسي دي جلوكوز برضه ممكن يصنع يعني بصوره يعني سينثيتيك او في المختبر عشان تو انهبيت ذا افكت اوف ذا هيكسوكاينيز 1. والدايكلورو اسيتيت برضه ممكن واحده من المركبات ممكن تصنع في المختبر او في الفارماسيوتيكالي، وتو بريفنت او تو انهبيت ذا افكت اوف ذا بايروفيت ديهيدروجينيز كاينيز انزيم. واللاكتو ايودو اسيتيت ممكن يعمل دي اكتيفيشن او انهبيشن لجليسرالدهايد فوسفات ديهيدروجينيز انزيم. لو ملاحظين ده كل الانزيمات دي بتعمل في الجلايكوليسيس باث واي، سواء كان انايروبيك ولا انايروبيك جلايكوليسيس. والتفاعلات دي كلها التارجت او الانزيمات دي تارجت ليه؟ لانه الكانسر سيلز دائما تكون more active than the normal cell في need of energy, so it will have more glycolysis than the normal cells. So if you can inhibit the glycolytic pathways in the cancer cells targeted by some inhibitors like these cases, you can reduce the rate of the cancer cell to split that rapidly. So you will reduce the rate of the cancer cells to propagate in the normal rate of the expected of the cancer cell. So some other type of the enzyme clinical use includes glucose oxidase, which is utilized for the measurement of the plasma glucose concentration or the glucose concentration. And the other examples, the use of the enzymes as tool of treatment of the injuries. For example, tissue plasmogene activator or the streptokinase is used the treatment of the acute myocardial infarction and so on. So these are examples of some enzymes that can be used in the genetic engineering to treat some of the lysosomal storage disorders, and these are the examples. Any question يا شباب؟ أي سؤال؟ يعني أنا هنا أخذت المحاضرة طويلة شوية لأنه جمعت كل الأشياء اللي عندها علاقة بالإنزيمز في المحاضرة اللي كان طالب مني أعمل محاضرتين منفصلات. محاضرة أولى عن الإنزيمز البروبرتيز والكلاسيفيكيشن والميكانيزم أوف أكشن، ومحاضرة ثانية عن الكاينetics والإنهبيترز، فضميتها في محاضرة واحدة وأتمنى أن تكون واضحة. ممكن لو ما في سؤال كده سيريس في السلايد ممكن توقفوا التسجيل يا باشمهندس طارق.